Hello, welcome to Biostudy Pathshala and I am Nilanjana. Today's topic is Gpcr, that is G protein coupled receptor, and it is the most important hot topic from the unit 4 of CSR UGC NET Life Science syllabus, that is cell signaling and cell communication. So let's get started. Before starting this topic, we have to know what is receptor. Receptor which receives signals or ligands. Okay? On the place of occurrence, these receptors is mainly of two types. These are intracellular receptor and cell surface receptor. Intracellular receptors mainly found inside the cell, that means in cytoplasm, right? And it is found mainly in soluble form, while cell surface receptors mainly found on plasma membrane, while it spans the plasma membrane like this way. And these cell surface receptors are mainly of three types. These are ion channel linked receptor, enzyme linked receptor, and Gpcr. Acetylcholine in case of nervous system shows ion channel linked receptor pathway, while receptor tyrosine kinases, tyrosine kinase associated receptor, and serine threonine receptor kinases show enzyme linked receptor pathway. Gpcr shows some examples like vision system, cyclic AMP pathway, and IP3 DAC pathway, and GABA receptor pathway. Alright, so today's topic is Gpcr. We will focus particularly on this topic. So you can see in this diagram, this area is for plasma membrane. Alright, and this blue helical structure representing the Gpcr. You can see this blue helical structure spans this plasma membrane 7 times. 1, 2, 3, 4, 5, 6 and 7 times. That is why this receptor is also known as serpentine receptor. And this serpentine receptor shows 4 loops. Loop 1, loop 2, loop 3 and loop 4. The G protein associates with this loop 3 area, while ligand shows the binding area with this particular area. After binding with the ligand, the Gpcr being activates, and after being activated, it activates further G protein. Now, this G protein shows 3 subunits. First one, as you can see in this figure, alpha subunit, gamma subunit and beta subunit. You can see this alpha and gamma subunit anchored with the plasma membrane, while this beta subunit flanked between this alpha and gamma subunit. Okay, now we are having a look on this particular note. Alpha subunit shows plasma membrane bound state and gamma subunit also shows the plasma membrane bound state. Now alpha subunit shows Gtps activity. So what is Gtps activity? Gtps activity is the formation of the GTP from Gtp. Alright, and GAP activity is Gtps activating protein activity. That means the activity or the enhances the activity of the Gtps. Alright, and this alpha subunit can be found either in GTP bound state or GTP bound state. This GTP bound state is always inactivate, while GTP bound state is active form. Alright, this G alpha subunit shows again 4 kinds of subsets. Okay, these are GS alpha subunit, GI alpha subunit, GQ alpha subunit and GT alpha subunit. This GS alpha subunit shows Adenylate Cyclase pathway. GI alpha shows GABA receptor pathway. GQ alpha shows PLC pathway and GT alpha shows vision system pathway. Alright, now we will start the Gpcr signaling pathway. It is the generalized view of the Gpcr signaling pathway. You can see here 6 steps are observed. Okay, in first step, the ligand binds to the Gpcr transmembrane receptor, and after binding with this ligand, this receptor protein being activated. After being activated, it interacts with the GDP bound state of the G protein, and it activates the G protein by associating of the GTP and dissociating of the GDP. After associating of the GTP with the alpha subunit, it goes for the inactivate effector and interact with this inactivate effector and resulting in the activation of the effector, and this effector further shows the responses, and after these responses this alpha unit shows its Gtps activity, and after showing this Gtps activity, this Gtp hydrolyzes to form one molecule of inorganic phosphate and the GDP. This GDP bound state of the alpha again becomes inactivated and reassociated with the beta and gamma subunit to form the inactivate G protein again. So that is how the G protein coupled receptor actually works. Okay. Now we will see the specific case of the Gpcr, like adenylate cyclase pathway. It is AC pathway, that means adenylate cyclase pathway. Alright, and here GS alpha subset involved in this particular pathway. Okay, after binding with this ligand, this Gpcr being activated and it further activates the G protein, and this G protein now reassociated with GTP. This GTP bound state of the G alpha S now activate the adenylate cyclase. This activated adenylate cyclase now forms the CMP from ATP. Alright, that means the level of the CMP now is high inside the cell. This high level of CMP now activates the protein kinase A. Now how this activation works? Okay, this protein kinase A shows 4 subunit. 2 is regulatory subunit and 2 is catalytic subunit. This regulatory subunit shows 4 sites where the CMP can bind, and after binding this CMP, the regulatory portion can dissociate from the catalytic portion and the protein kinase A become activated. Now this activated protein kinase A transports into the nucleus, and this activated protein kinase A activate the inactivate CREB, and the full form of the CREB is CAMP, response element binding protein. Now this CREB is being activated by the presence of activated pKa. Okay, now this CRAB binds to CBP, that is CREB binding protein, that means CREB binding protein. Now this CREB binding protein and CREB complex, that means CREB plus CBP complex, binds to CRE portion, that is CREB response element portion, and transcription on and further responses are observed. So that is how Gpcr signaling pathway in case of adenylate cyclase system can be observed. Alright, this is the summary of the adenylate cyclase pathway. Okay, and the binding of the ligand to the receptor, you can see that it resulting the formation of the active alpha subunit with GTP bound state. Now this GTP bound state of the alpha activates the adenylate cyclase and shows the response further, and after the response the alpha unit shows its GTPS activity, which dissociates the GTP in the form of GDP and inorganic phosphate. Now this GTP bound state again becomes inactivated and reassociated with the beta and gamma subunit, and again this pathway becomes inactivated. So this is how Gpcr system works, and I hope this brief session on the Gpcr will help you a lot. Thank you for watching this video, and if this video is helpful for you, make sure to share this video with your friends and students, your known circle, and lastly don't forget to subscribe this channel and hit the bell icon below. So till then, be safe and be healthy. Bye bye.